Welcome to the Deep Dive. Today we are buckling up for a really deep examination of what might be the most profound technological shift happening right now.
Absolutely.
We're talking about the collision of artificial intelligence and, well, the fundamental building blocks of life itself. The pace of change in biotechnology isn't just fast anymore, it feels geometric.
It really does. And I think our mission today is to take this whole stack of sources we have, which blend high stakes financial projections, we're talking 2026 outlooks, with these bleeding edge reports straight from the lab.
Right, from the clinical trials themselves.
Exactly. We need to figure out what the core transformation is. This deep dive is about uncovering those key breakthroughs in genetics, in medicine, synthetic biology, and, really understanding the catalyst driving it all.
And that catalyst, it's unavoidable, isn't it? It's AI.
It's beyond.
AI is no longer this tool that's just for optimizing code or crunching stock market data. It's fundamentally redefining biological discovery. It's like a force multiplier for every single scientist working today.
The synergy is just so immediate, so pervasive. I mean, the financial sources we looked at are crystal clear on this.
What do they say?
They're forecasting that AI adoption is going to translate into measurable productivity gains. That it's going to modestly raise what they call total factor productivity projections across the entire U.S. economy.
Especially in healthcare.
Especially in healthcare. AI is taking these processes that, historically took years of painstaking wet lab labor, and it's turning them into computational problems that can be solved in weeks or even days.
Okay, let's unpack that phenomenal acceleration right away. Let's dive into our first section, redefining medicine. Because we're seeing clinical trial results right now in early 2025 that gene editing has, it's moved way beyond just a theoretical promise.
Oh, absolutely. It's now a precise, measurable clinical reality.
And that's the critical difference, isn't it?
It is. The precision of gene editing is just improving so dramatically,
Yeah.
and the results are showing up in the data for both extremely rare disorders and, much more common persistent diseases.
Let's start with a rare disorder. That's usually where the highest degree of precision is needed. Tell us about this company, Biotech Therapeutics.
Biotech reported some very, very promising results in early 2025. They're targeting a condition called primary hyperoxaluria type 1.
Okay, what is that?
It's a severe inherited kidney disorder. Basically, a genetic flaw causes the body to overproduce a substance called oxalate. This leads to incredibly painful kidney stones and eventually catastrophic renal failure.
So what was their approach, and more importantly, what was the impact?
They used an in vivo gene editing approach.
Meaning it happens inside the patient's body.
Exactly, directly inside the body. The therapy successfully reduced those harmful oxalate levels by nearly 70% in the patients they tested.
70%? Wow.
I mean, that is a massive clinical result for a disease that, before this, required just constant complex management and often eventually an organ transplant.
A 70% reduction is absolutely life-changing for those patients. But the delivery mechanism, that's just as revolutionary as the edit itself, right?
Absolutely. The therapy relies on these tiny fat-based particles. You've probably heard of them. They're called lipid nanoparticles, or LNPs.
The same tech from the mRNA vaccine.
The very same.
Oh.
LNPs are basically microscopic delivery trucks. They safely wrap up the gene editing machinery, shield it from the immune system, and, this is the crucial part, they're engineered to go to specific cells.
So in this case, the liver.
In this case, the liver, which is where that faulty enzyme is produced. This incredibly precise non-viral delivery method is really the key to making systemic gene editing both viable and safe.
So we've taken a huge step towards solving a devastating genetic disorder. Can this kind of precision tackle broader challenges like, say, chronic viral infections?
We're seeing some very promising signs that it can. You have to look at the work being done by a company called Precision Biosciences.
Okay.
They're developing a therapy that's aimed specifically at eliminating the persistent chronic reservoir of the hepatitis B virus, HBV.
And current drugs, they just suppress it, they don't cure it.
Exactly. Current medications can suppress HBV, but they very rarely clear it completely. So patients often need lifelong treatment, and they still face a continued risk of liver cancer.
So what did their early trials show?
The early data showed substantial reductions in viral markers in 2 out of the 3 patients they tested. And this was even at the lowest dose they tried.
That's very encouraging.
It's highly encouraging. The goal here isn't just suppression. The goal is a functional cure by targeting the virus's latent DNA that's hiding inside the liver cells.
So you're moving from just managing a chronic disease to potentially wiping it out of the body for good.
Eradicating it altogether. That's the ambition.
And this is where we really bridge that gap. Because all of that medical success relies on these increasingly sophisticated molecular tools. And as we said at the top, AI is now designing those tools completely from scratch. Tell us about OpenCRISPR-1.
OpenCRISPR-1 is, it's being heralded as a landmark achievement. It is the first entirely AI-designed CRISPR editor.
So historically, we've had to find these systems in nature, in bacteria, and then just try to adapt them.
Right. We'd find them and then optimize them in the lab. OpenCRISPR-1 just breaks that entire mold. It's a designer Cas9-like nucleus with a fundamentally novel sequence.
Okay, what does that mean? A novel sequence. How is that different from what we used to do, like directed evolution in a lab?
Well, traditional methods, even something as advanced as directed evolution, are sort of inherently limited by what already exists or by making tiny incremental changes to existing structures.
You're stuck in a certain design space.
You are. AI, on the other hand, especially these vast generative models, can explore a far, far larger chemical and biological space all at once. It uses deep learning to understand the fundamental rules of protein folding and binding, and then it generates completely new sequences that follow those rules but achieve, superior performance.
Things like greater stability or higher specificity.
Exactly, and things that nature just maybe hasn't had a reason to evolve yet. The AI is inventing a better, more precise solution.
And what were the specific performance gains for OpenCRISPR-1? Did it work?
It did. It offers performance that's comparable to, and in some cases even better than, the gold standard, the natural STCas9 system.
But the real insight, I imagine, is specificity.
That's the key, enhanced editing specificity. This is so crucial because it suggests the potential for significantly reducing unwanted side effects.
Those off-target effects you always hear about, where the editor cuts DNA in the wrong place?
The very same.
Yeah.
Minimizing those is the holy grail for safety in gene therapy.
And on top of all that, it's open source. What is the real-world impact of an AI-design, open-source gene editor being available to everyone?
It dramatically democratizes the field. Think about it. If you are a small biotech startup or a university research lab, or even a cell and gene therapy developer in a developing country,
Right.
access to high-performance, validated tools without these restrictive licensing fees, it just lowers the barrier to entry immediately.
So research can accelerate everywhere all at once.
In a distributed way. It ensures that technology doesn't just benefit the few, but it really fosters global innovation in medicine.
That transparency and accessibility, that's a powerful lever. Okay, let's transition from gene editing to the other major revolution, mRNA therapeutics. For years, mRNA was just synonymous with vaccines.
That's all anyone thought of.
But our sources show that by 2025, it's really matured into a general therapeutic modality. It's tackling a much wider spectrum of conditions now.
It really is the ultimate biological software. You can instruct the body's own machinery to produce basically whatever protein or antigen you can code for. And the most compelling recent data is coming out of personalized cancer vaccines.
Let's zoom in on that. The phase 2b Keynote 942 trial. This involves the personalized mRNA cancer vaccine from Moderna and Merck, mRNA 4157, combined with Keytruda. What did that trial show for high-risk melanoma?
So this trial focused on patients with high-risk stage 3 or IV melanoma. These are patients who had already had their tumors surgically removed. The big challenge for them is preventing the cancer from coming back.
Preventing recurrence.
Exactly. When they added the personalized mRNA vaccine to the existing standard of care, Keytruda, the combination showed a clinically meaningful and statistically significant reduction in the risk of the disease recurring or death.
Can you quantify that impact for us? What does significant mean here?
It was a nearly 44% cut in the risk of recurrence.
Wow.
And you have to put that in perspective. This is not some small incremental improvement. This suggests that the mRNA vaccine is successfully teaching the patient's own T cells to recognize and hunt down those microscopic residual tumor cells that surgery might have missed.
It's truly personalized, targeted immunotherapy.
Yeah.
And it's working against one of the most aggressive forms of cancer.
It is.
That's astonishing for a personalized approach.
Yeah.
But this technology is also showing promise in off-the-shelf treatments too, isn't it?
Correct. You don't always need full personalization. BioNTech, another leader in this space, showed an 18% response rate with their off-the-shelf melanoma vaccine, BNT101.
And this was in which patients?
Specifically in refractory patients. These are people who had previously failed to respond to other treatments. It just highlights the versatility of the platform.
So beyond cancer, How is mRNA broadening its horizons? Where else are we seeing these revolutionary applications pop up?
The potential is just vast, especially in the rare disease space. I mean, think about protein replacement therapies.
Okay.
Instead of manufacturing a missing enzyme in a factory and then injecting it into a patient every week, you could use an mRNA therapeutic to encode that missing enzyme.
So the patient's own cells become the factory.
Their own cells become the factory, producing the protein they need endogenously.
Yeah.
This could be for metabolic disorders like PKU or certain lysosomal storage diseases. It just shifts the entire burden of treatment.
From a chronic drug infusion to a more natural, sustained biological function.
Exactly. And we're also seeing great potential for regenerative purposes. You can instruct cells to produce factors that induce tissue repair after an injury, or even target specific stem cell populations to promote localized regeneration.
The challenge there, I assume, is delivery.
It's always delivery. Ensuring safe, repeated, and highly targeted delivery is the key.
Which brings us back to that point. The success of mRNA beyond the pandemic really depended on improving stability and targeting. How have those advancements materialized by 2025?
Well, the early logistics hurdles, the need for that ultra-cold storage, have been largely mitigated.
Thank goodness.
Advances in the mRNA chemistry itself have significantly improved the stability of the molecule. So you're looking at much more manageable refrigeration requirements now, which, vastly expands its global deployment capability.
And what about the LNPs, the delivery trucks?
They've been highly refined. We can now target organs beyond just the liver. We're seeing them engineered to go to the spleen, muscle tissue, and increasingly specialized immune cells. This just expands the therapeutic reach across the entire human body.
Okay, so we've established AI is designing better gene editors. It's helping refine complex therapeutics like mRNA. Let's move to section 2, which positions AI not just as a tool, but as a genuine co-scientist in the lab.
A partner.
A partner capable of massive, independent intellectual leaps.
And this really begins with structural biology, which is, the foundational science of understanding how all these biomolecules actually work. The massive leap forward here is AlphaFold 3, which was released in late 2024.
AlphaFold was already revolutionary for predicting single protein structures. Why was version 3 seen as such a major turning point?
Because it shifted the complexity. It went from modeling a single element to modeling an entire dynamic system.
So if previous versions were like defining a single word in a biological dictionary.
A brilliant definition, but still just one word. AlphaFold 3 moved beyond that. It can now model the entire complex dynamic sentence. It can model the interaction.
What does that mean in practice?
It means it extended its modeling from just isolated protein structures to predicting the structures of complex assemblies, how multiple proteins bind together, and crucially for drug design, modeling precisely how proteins interact with DNA, RNA, and small drug molecules.
It's modeling the internal dialogue of the cell.
That's a great way to put it.
And how accurate is this digital prediction now? Is it reliable?
It achieves substantially improved accuracy. Our sources indicate it's at least 50% better in predicting some of these biomolecular interactions compared to any previous methods.
So a drug designer can see potential binding sites on a computer screen before ever stepping into a lab.
They can see it all in silico.
Yeah.
This could save millions of dollars and years of work.
That reduction in friction and cost is key. And you mentioned accessibility before. This isn't just for, elite academic institutions anymore, is it?
Not at all. By 2025, the public server and code release for AlphaFold 3 made it widely accessible to researchers and students all over the world. It has fundamentally transformed structural biology. It used to be this complex bottleneck discipline requiring huge, expensive equipment.
X-ray crystallography and all that.
All of that. And now it's becoming a routine computational discipline you could do on a laptop.
And that acceleration is now delivering tangible results. The ultimate proof point came in 2025. The first entirely AI-designed antibody entering human trials. That feels like a true watershed moment.
It absolutely is. This is Absci Corporation's drug, ABS-101. It targets a cytokine called TL1A, which is a known player in inflammatory bowel disease, or IBD.
And it's in phase 1 trials now.
It entered phase 1 in mid-2025, which officially made Absci a clinical stage AI drug company.
But designing a biologic entirely with AI feels like a massive computational risk. The immune system is so complex. How much confidence can we really have in an AI optimizing a drug in silico before we even have phase 1 safety data?
That is exactly the right question to be asking because the risk is enormous. But what's interesting here is that the AI optimization wasn't focused on efficacy first. It was focused on manufacturability and patient convenience.
Okay, tell me more.
The AI was used to optimize the molecule across three key parameters at the same time, which is something that's very, very difficult for a human team to do using traditional methods.
And what were those 3 parameters?
First, achieving an extremely high binding affinity for the target. Second, designing for an extended half-life so the drug stays active in the body for longer.
Which means less frequent dosing for patients.
Right, it enables potential quarterly dosing. For a chronic IBD patient, that's a massive quality of life improvement over, say, biweekly or monthly injections.
And the third parameter?
The AI specifically engineered the antibody for low immunogenicity.
Meaning the patient's immune system is less likely to attack the drug itself.
Exactly. So the drug is less likely to fail over time. The AI isn't just finding new leads, it's generating patient-friendly, manufacturing-ready, superior quality drugs and doing it faster. It's fundamentally shortening discovery cycles.
Which means pharma companies might start licensing these high-quality AI-generated leads instead of relying only on their huge internal discovery pipelines.
It represents a seismic shift in how R&D is organized globally.
But AI's influence isn't just in designing molecules. It's also becoming this agentic partner in the daily work of the lab itself. Let's define agentic AI. What does that mean?
Agentic AI means the model is moving beyond just being a smart search engine or a data analyst. It's capable of taking a high-level goal, breaking it down into subtasks, planning the steps, executing them, learning from feedback, and adapting its plan to achieve the final objective.
So it's less like a calculator and more like a junior scientist you can delegate to.
A junior scientist with a PhD who you can give a complex assignment to, yes.
And we have hard data showing its intellectual capability has surpassed human experts incredibly quickly.
The jump is staggering. I mean, as recently as 2022, AI models were consistently underperforming human PhD-level experts on complex biology questions. By 2025, the frontier models have long since exceeded the human expert baseline.
By how much?
They're showing a performance gain of plus 0.6 relative to that baseline. And performance in complex chemistry is catching up just as fast.
What was the fundamental shift in the modeling architecture that allowed that huge leap in scientific reasoning in just a few years?
It's the shift towards what some are calling reasoning compute, like in systems such as OpenAI's O1. Instead of just scaling up the size of the model, these systems spend a lot more computational time generating a robust chain of thought or an internal plan before giving you the final answer.
So it's simulating logical deduction, not just pattern matching.
Precisely. That focus on structured, multi-step reasoning makes them fundamentally better at the complex, sequential tasks you find all over science and mathematics.
And this capability, again, democratizes complex tasks, like you mentioned earlier with the protocol writing.
Yes, so the data on protocol generation is really illustrative. They found that non-experts who use these frontier AI models to write complex experimental protocols for something like viral recovery had 4.7 times higher odds of writing a feasible protocol.
One that could actually work in a lab.
Right, compared to a control group that was just using Google and textbooks.
So the AI is effectively acting as a senior lab manager, ensuring basic experimental feasibility and doing safety checks, even for a novice.
Exactly, and the industry is now formalizing this. Google's AI co-scientist, powered by Gemini 2.0, is designed specifically for this role-generating new research hypotheses, detailed overviews, and structured experimental protocols. Agentic AI is moving from answering your questions to actively collaborating with you.
And there's a projection that 75% of companies could be investing in this by the end of 2026, is creating these digital colleagues.
Right. It allows small teams to punch way above their weight, letting the AI handle complex data synthesis while the human experts focus on strategy. It's the ultimate productivity lever for the intellectual workforce.
Which is why all that investment capital is flowing so aggressively into the infrastructure that supports it.
It is.
That is a perfect segue into our third section, synthetic biology. This is where the revolution moves beyond the human body and starts re-engineering our entire world: materials, agriculture, manufacturing, even data storage.
Synthetic biology is really the intentional combination of engineering principles with biological systems. It's about designing and building new biological parts, devices, and systems, or redesigning existing natural ones for a useful purpose.
We're literally programming genetic circuits into living organisms.
We are. So those big fermentation tanks you see in industrial production, they're starting to replace chemical plants. Living cells are becoming the new microscopic factories.
For industrial use.
Exactly. We're seeing novel organisms coming out of labs, microbes, engineered to be incredibly efficient industrial workers. They're being designed to make advanced biofuels, complex pharmaceuticals that are hard to synthesize chemically, and biodegradable plastics.
And you can use them for environmental applications, too.
Yes. CRISPR-modified microbes are being tested for large-scale carbon capture or for rapidly degrading complex pollutants in the environment.
And this convergence is now manifesting physically through the bioprinting boom. What are next-gen bioprinters creating today that we couldn't make just a few years ago?
We're moving way beyond simple single-cell constructs. The next-gen printers are creating complex living tissue with multiple cell types, all layered and structured to mimic actual biological function.
And this is already being used in clinics.
It is. It's being used to create custom-printed skin grafts for severe burn care and custom bone implants that are tailored exactly to a patient's anatomy.
And the immediate goal in medicine here is to eliminate immune rejection, isn't it?
That is the holy grail. Major hospitals are actively forming partnerships to set up dedicated bioprinting facilities that can produce patient-matched tissue grafts on demand.
So you use the patient's own cells as the ink.
You use their own induced pluripotent stem cells as the starting material. You differentiate them into the tissue you need, and you drastically reduce the risk of the body rejecting the graft. It solves the major complication of traditional transplants.
That is a practical, life-saving application. But let's go to the wild edge of this convergence, because it's really starting to blur the lines between what is alive and what is engineered. Tell us about these programmable biological robots, the xenobots.
Xenobots are just fascinating. They're self-assembling, programmable biological robots, and they're constructed from living cells, often frog cells.
Yeah, what do?
They've demonstrated the ability to self-propel, to carry tiny payloads, and even to self-replicate in a very rudimentary way. They really represent the frontier of engineered, living, programmable matter.
And we are also seeing the literal integration of technology inside living organisms, which sounds like something straight out of a cyberpunk novel.
It's happening right now. Research from Lancaster University successfully demonstrated 3D printing glowing shapes and, more importantly, conductive circuits inside living nematode worms.
Wait, printing electronics inside a living worm? How is that even possible? And what's the practical application?
They used a very precise photonic 3D printer and specialized inks. The practical application, the ability to interface with biology so intimately, suggests incredible potential for improving internal electronic implants.
Like pacemakers.
Exactly. Traditional implants like pacemakers or bionic ears, they face issues with infection and long-term integration with soft tissue. By printing or growing the conductive material within the biological host or using these bio-friendly soft materials, you could reduce those issues and lead to much more durable, less invasive electronic medicine.
Shifting now from tiny integrated circuits to the biggest data problem humanity faces, DNA data storage. Given the exponential rate at which we're generating data, why is this biological solution becoming a necessary alternative?
It's a crisis of capacity, plain and simple. Sources are projecting that by 2030, global digital storage demand is going to exceed our supply by a staggering 20 million petabytes.
And our current hard drives and magnetic tapes just can't keep up.
Not sustainably. DNA offers two immense advantages: density and longevity.
How dense are we talking?
Theoretically, a single gram of DNA can store an exabyte of data.
An exabyte.
You could store the entirety of the world's current digital data in a structure about the size of a sugar cube. And the longevity is equally profound. Stored DNA can last for thousands of years without degrading, unlike a hard drive that might fail in a decade.
So how do you translate ones and zeros into A, T, C, and G?
You encode the digital data into DNA sequences using chemical synthesis. You assign binary pairs to the 4 nucleobases. For example, 00 could be A, 01 could be T, and so on.
And machines just build these DNA strands.
Synthesis machines build these specific strands. The future is moving towards semiconductor chips and enzymatic synthesis, which will dramatically improve the scale and efficiency and make this a real commercial infrastructure component.
Finally, let's look at sustainable disruption. How is bioinnovation changing traditional supply chains in things like food and manufacturing?
Starting with food, cultivated coffee, which is grown in bioreactors from coffee bean cells, is a massive potential disruptor. The environmental benefits are startling. This approach requires 95% less water than traditional farming and completely eliminates the need for pesticides.
And what's the environmental cost of traditional coffee right now?
Traditional coffee farming is a major driver of deforestation. It consumes an estimated 250,000 acres of forest every year, often in critical biodiversity hotspots. Lab-grown coffee could fundamentally change that whole supply chain,
You could produce it year-round, anywhere.
independent of climate volatility and without the environmental footprint.
And beyond food, how is this changing core infrastructure materials?
We're seeing fascinating progress on lab-grown wood, where cells are cultivated to create custom wood products with enhanced properties like modified grain patterns or superior strength all without cutting down a single tree.
But the most durable disruption might be in concrete. Tell us about self-healing concrete.
This is a fantastic innovation. It harnesses specific strains of bacteria. When a crack forms in the concrete, water seeps in and activates these dormant bacterial spores.
And the bacteria wake up and go to work.
They wake up, they metabolize nutrients that are already in the concrete, and they produce calcium carbonate limestone, which naturally fills and seals the crack.
How effective is it?
It's been shown to fill cracks up to 0.46 millimeters wide, which is 4 times more effective than traditional chemical methods. This could significantly enhance the resilience and lifespan of massive infrastructure projects like bridges and tunnels.
Okay, now we have to enter section 4, the ethical and governance nexus. Because with science moving this fast, we know governance always lags behind. It forces regulators to play catch-up with technologies that challenge our very definition of life.
That is the core conflict. And there's perhaps no area more ethically complex right now than synthetic embryo models, or SEMs.
For listeners who might not be familiar, what exactly are SEMs and what makes them such an ethical minefield?
SEMs are created using stem cells. Often they're induced pluripotent stem cells, or iPSCs, which are adult cells that have been reverted to an embryonic-like state.
So no sperm or egg is involved.
No sperm or egg. Scientists guide these cells to self-organize, and they recreate the very earliest stages of embryogenesis in vitro. The ethical complexity comes in because they can develop structural similarities to natural embryos, forcing us to draw new lines on what constitutes life worthy of protection.
What are the immense scientific insights that justify walking this ethical tightrope?
They offer unmatched insights into early human development. This period, days 1 to 14, has long been a complete black box for science.
So SEMs let us see inside.
They let us model genetic diseases, understand why implantation fails, and study the fundamental processes of how our organs begin to form, like how the human heart starts developing. This knowledge is crucial for regenerative medicine.
And they can generate specific cell types we need for therapy, right?
Yes, critically they're being used to generate PGC-like cells, the embryonic precursors to sperm and egg cells. Studying how SEMs generate these cells gives us unprecedented insight into the causes of germ cell cancers and human infertility.
And you can make them patient-specific.
You can. You can derive them from a patient's own iPSCs, which allows researchers to test drug safety and efficacy on models that closely reflect that patient's specific genetic makeup. It's a huge step toward truly tailored medicine.
However, the creation and use of this material, even if it's non-living and in a dish, requires incredibly stringent ethical oversight.
Absolutely. Institutions like the Sanger Institute, which might use this material for research, have to operate under strict Research Ethics Committee, or REC, approval. These RECs scrutinize every application, focusing on necessity, scientific merit, and adherence to established governance.
And the field is moving so fast that those rules need to be transparent and international.
They have to be. We need transparent control systems to manage the evolving ethical, legal, and regulatory questions.
And there's a long-term goal that these models, ironically, will actually reduce the overall ethical burden in research.
That is the explicit hope. As our foundational knowledge grows from these advanced models, the reliance on traditional embryonic and fetal samples for research is expected to decrease significantly.
And the ultimate destination.
The ultimate destination, fueled by AI and these massive datasets, is to move toward fully in silico, entirely computational models of development, which would eliminate the need for physical biological matter altogether.
Okay, let's look at regulation trying to catch up to AI and gene editing. Specifically, the security risks. What are regulators most concerned about with agentic AI?
The misuse risk is paramount. On the pure software side, we already have documented evidence that AI can spontaneously self-replicate and evolve in code.
And on the biological side?
The concern is that AI now has the capability to generate proposals for novel toxic compounds or to optimize delivery systems for harmful materials. This presents a clear and present security risk. Regulatory oversight has to evolve rapidly to prevent sophisticated misuse.
And that immediate concern is a core driver for the strict new policies we're seeing, particularly out of Europe.
Yes, the EU AI Act is a global watershed moment. Our sources highlight that this act has fundamentally reshaped biotech and healthcare globally.
Why? What's its main mandate?
It mandates unprecedented transparency, accountability, and regular algorithmic audits, specifically for what it defines as high-risk applications.
That sounds like a massive infrastructural lift for companies. What qualifies as high risk, and what does compliance actually require?
High-risk examples include things like AI-guided robotic surgery, diagnostic algorithms, or AI used in clinical trial recruitment. Practically, the Act mandates that companies must document every single decision-making step their AI takes.
So it's not enough for the AI to get the right answer.
No. The company has to prove how the AI got the right answer and that the process was fair and unbiased. They have to prove with detailed records that their AI systems avoid demographic bias in genomic analysis, for example.
So for a big company like Roche or Siemens, this means hiring specialized audit teams and building entirely new compliance systems.
It does. It means investing heavily in that infrastructure. The goal is to ensure that while AI accelerates discovery, the core principles of scientific integrity, safety, and non-discriminatory practice are prioritized over raw speed. It slows things down a bit, but it ensures trust in the results.
And on the purely biological side, the global regulation of human gene editing, especially CRISPR, is also intensifying.
There's a universal tightening of regulations across all the major jurisdictions: China, the US, the EU. The focus is on drawing a very firm line between therapeutic gene editing and germline editing.
Let's clarify that distinction for the listener.
Therapeutic gene editing, or somatic editing, targets non-reproductive cells to treat a patient for a condition like sickle cell disease. These edits are not passed down to future generations, and they're widely prioritized.
In germline editing,
Germline editing modifies reproductive cells, which means the changes are heritable. They get passed down. Governments are severely restricting or outright banning non-essential germline modifications to prevent irreversible consequences and to protect the principle of informed consent across generations. It's about balancing medical progress with profound social responsibility.
these technological shifts don't happen in a vacuum. They require immense capital. Let's move to our final section: investing in transformative technologies. This is the financial community placing its bets on this revolution.
And from a macro perspective, the investment community sees these technologies as the crucial growth engines that will dictate national competitiveness. As we noted earlier, AI is forecast to translate into measurable productivity gains, raising total factor productivity, or TFP.
Let's ground that concept, TFP. It sounds a bit abstract.
TFP is essentially a measure of economic efficiency. It's how much output growth you get that isn't just from adding more labor or more capital. It measures innovation.
So if you automate a lab protocol with AI, one scientist can produce 5 times the data they could before. That gain is TFP.
Exactly. Without needing 5 times the equipment or 5 extra years of schooling.
And why is boosting TFP so essential for advanced economies right now?
Because advanced economies are facing major demographic headwinds. We have rapidly aging populations, fewer working-age people supporting more retirees. The TFP boost from AI, particularly in high-leverage sectors like tech and healthcare, is seen as crucial to sustaining long-term growth without relying on population increases.
Technology has to solve the structural labor shortage.
In essence, yes.
So investors are chasing that efficiency and growth. This explains the massive concentration of capital, particularly in private markets, focusing on this AI biotech nexus.
The numbers are stark. AI investment has accounted for over 60% of all venture capital investment over the last 12 months.
60%.
That flow reflects a clear conviction that the majority of future economic value will come from these platform technologies, the AI engines, the LNP delivery systems, the synthesis platforms that enable the entire biotech revolution.
And private equity firms are following this trend, but with their own strategies.
Absolutely. PE firms are increasingly focused on businesses tied to tech and AI, but they're deploying more sophisticated strategies. They're moving beyond simple buyouts and are focusing on acquiring smaller, specialized businesses, what they call add-ons, to enhance the technical capabilities of their core platform investments.
Dazzling expertise.
Bundling AI and biotech expertise, it's a key value creation strategy right now.
Now, let's talk about hedge funds, which thrive on volatility. How are the current market shifts favoring active strategies that specifically target biotech?
We've entered a new market regime. It's characterized by elevated volatility, higher equity market dispersion, meaning individual stocks are behaving more independently, and higher interest rates. These conditions are tough for passive investors, but they create a much more dynamic environment for alpha generation.
Okay, for someone who isn't a hedge fund manager, what does alpha generation actually mean here?
It means generating returns that are independent of the broader market's movements.
And biotech is an inherently volatile sector, right? Full of these binary outcomes like a clinical trial succeeding or failing spectacularly.
Exactly. And biotech is called out as a specific source of opportunity because of those binary events. Active hedge fund managers, with their specialized knowledge, can position themselves effectively around key clinical milestones or regulatory announcements. The increased dispersion means a successful biotech stock can surge, even if the broader market is flat, allowing them to capture those outsized returns.
And naturally, just as AI is transforming the lab, hedge funds are integrating AI into their own processes to get an analytical edge.
It's a complete transformation of their investment process. They're leveraging large language models for everything from improving coding and operational efficiency to actually generating alpha and refining their investment theses.
How so?
An LLM can rapidly analyze thousands of clinical trial reports, scientific patents, and regulatory filings all at once. It can structure that information and identify subtle connections that a team of human analysts might miss, allowing them to formulate systematic, actionable investment strategies much, much faster.
So the sophistication of the investment decisions is rising right alongside the complexity of the science being invested in. It sounds like a new era for active management.
It really does, especially since the traditional diversification you got from a stock and bond balance has become less reliable lately. That rising, unstable correlation between stocks and bonds is a major challenge. So the role of hedge funds is shifting. Their ability to generate these idiosyncratic returns, independent of the market, positions them as a critical risk moderator for large institutional portfolios when both equities and bonds might be falling at the same time.
So the complexity of biotech, driven by AI, is creating both unprecedented medical progress and this dynamic, high-stakes new frontier for capital.
It confirms that we're in a period defined by these competing global forces. On one hand, you have powerful constraints, economic nationalism, policy fragmentation, tighter trade.
And on the other hand?
On the other, you have this incredible broadening technological momentum from AI and biotech, coupled with immense public and private investment targeting these fields. The tension is palpable.
And that technological momentum is what links a high-level policy debate, like the EU AI Act, directly to the integrity and speed of scientific discovery on the lab bench.
Precisely. That regulatory framework ensures that as AI accelerates the design of gene therapies and diagnostics, the core principles of transparency and accountability are non-negotiable. The speed of innovation is being coupled with necessary ethical checks.
Okay, let's end with the most fascinating and provocative implication from our sources about where this AI biotech convergence is ultimately leading us. If the goal is to make discovery faster and cheaper, what's the final destination for scientific R&D?
The ultimate destination, the intellectual terminus of combining AI and biology, is the radical reduction in our reliance on physical samples and traditional wet lab work altogether.
So all the knowledge we're gaining from physical samples now, like human embryo atlases.
That's the foundational data. The combination of AI and these massive data sets allows that foundational understanding to be scaled exponentially.
The explicit goal, then, across all of advanced biological research,
is to generate fully in silico models. Computational models of cells, tissues, organs, and eventually entire biological systems. Imagine having a perfect digital twin of a healthy or a diseased human cell that accurately reflects in vivo conditions.
What would that ability unlock? What could you do?
You can make sophisticated, highly accurate predictions about disease progression, drug efficacy, toxicity, and therapeutic design all digitally, without ever having to synthesize a compound, inject an animal, or conduct a costly, time-consuming physical trial.
This fundamental shift from the wet lab experiment to the predictive digital experiment.
It will change scientific R&D forever. It will accelerate discovery in ways we're only just beginning to grasp today. The ultimate lab very soon will exist entirely on a hard drive.